let's learn some replication. So, replication. We've been learning about DNA. We've been learning about who discovered DNA, the process of creating DNA, and, well, not creating DNA, discovering it, the experiments that led to it. And now we're going to learn about how DNA makes a copy of itself. So this process is called replication. Now, DNA doesn't just magically appear. In order to get new strands of DNA, it needs to come from an existing DNA. And this is the process called replication. Each strand of DNA gets broken in half, and it uses half of an old strand as a template, and then half of a new strand is created from that template, and it kind of reads it. So that's what we're going to learn about. It is really quiet in here today. It's so unusual.
I don't like it.
You don't like it? It's not so bad. It's nice. Oh, I forgot to tell you, I got permission to bring Zozo.
The bird, not the snake.
bird, Zozo. So Zozo is approved to come to school, and I will bring him not this coming week, but the week of finals. That Monday is a normal day. It's not a block schedule day, so that day we'll just be reviewing for our test, and so he will come that Monday.
When is that test, by the way?
I
I have to schedule for FBLA finals.
I will tell you as soon as we're done with notes, because the calendar is on my iPad. I can iPad recording. So. Alright, so as replication begins, the enzyme helicase breaks the hydrogen bonds that hold the double helix together. So helicase is an enzyme. Remember, anything that ends in ASE is an enzyme. And think of it as kind of like a zipper. It's going to unzip the DNA strand. So it unwinds it and it unzips it. And these hydrogen bonds are going to break apart. And the two DNA strands are going to separate. So you've got nucleotides on one side, nucleotides on the other. And the helicase is basically just coming through and breaking those hydrogen bonds. And again, that's why I said those hydrogen bonds were important, because they're easy to break, but they're also easy to reform. so there are these things called primers. The primers are these short little chunks of DNA sequence, nucleotide sequence, and they kind of start and tell the process where to start, and then they signal for DNA polymerase, another enzyme, the enzyme ASE, and they're like, okay, we need to start here, and you can start reading here. And the DNA polymerase comes along and it starts putting the other side together. So both strands are getting copied at the same time, and the DNA polymerase is reading the template and matching everything up. So the nucleotides, there's like free floating nitrogen bases, the adenines and guanines and stuff, they're coming in and getting matched up to their pairs, and they're being matched up by the DNA polymerase. So the helicase is splitting it apart, the primers are in place, and then the DNA polymerase are reading the strands and matching everything up as it goes along. And both strands are having the replication take place at the same time.
Is the final harder than all the other tests?
It's longer. I don't know if it's harder. Depends on how much you remember from the other tests.
Are the same questions from the test only?
Mm-hmm.
Am I still allowed to open up the old test and look at the answers?
I'll probably let you on the review days. Alright, so nucleic acid hybridization, basically what that means is the hydrogen bonds are going to form kind of spontaneously between the nitrogen bases. So it's kind of like a magnetic pull almost. They can't help but, the adenines and the thymines can't help but like be pulled together, and the same thing with the guanines and the cytosines. They can't really help but be spontaneously pulled together. So those hydrogen bonds just automatically get attracted to each other between the two different bases. So when they are separated by helicase, that's unnatural. But when DNA polymerase comes and reads them and brings them back together, they hybridize spontaneously, and that hydrogen base just kind of magnetically pulls them together, and that hydrogen bond forms. So this is a part of, this is called DNA synthesis. Synthesis means to create. Now we will do protein synthesis, like I said, when we get back from Christmas break.
The other biology class had a test today,
Mhm.
and they did it like what our first test was. That was their test. They kept asking what was, and I could
that's good.
Somewhat.
So you're learning them.
But I don't know what about the test.
It sounds like you have test anxiety.
I can never get tests. I used to fail every single test. Like my first English test, I failed.
So test anxiety is a actual thing that people can do really good, like studying and preparing, and then when they get to the test, they bomb the test. It does suck when you get to a college class though, because the majority of college classes have weighted tests. Alright, so enzyme DNA ligase, so another enzyme, we have helicase, DNA polymerase, and now we have ligase, so 3 different enzymes are being used here, is going to come up behind and then seal in any gaps. So by the time that we are done building our strand, we need another DNA to come in and kind of seal everything up, and that's what ligase does. Ligase is going to come and make sure there aren't any gaps left over, and if there are any gaps, it's going to seal everything up and zip it up nice and tight. So helicase is going to break it down, polymerase is going to kind of read it and make sure everything is matched up, and then ligase is going to seal it all up and make sure everything is nice and neat. For lunch,
Sausage pizza.
sausage pizza.
I hate their sausage pizza. No offense to their sausage pizza. It's just I don't like it. There's something wrong with the cheese. No, we're supposed to go to the gym because it's some games going on. I asked him if I could max out today, and he said, "No, we have this going on." Something like he said state.
Football state game that he's going to. Isn't it?
It's not our school. Why is he going?
So both of the 2 strands of the parent molecule are being copied at the same time. This is a semi-conservative replication. So if it was fully conservative, then both strands would be the original. So that's not the case. Semi-conservative because 1/2 of the strand is old and 1/2 of the strand is new. So they're conserving the old half from the original parent one. The original parent one, 1/2 of it is being used in the new strand. So it's a semi-conservative process. And we'll look at some pictures and we'll watch a couple videos that explain the replication process, and it'll make a little more sense as we do that.
What are we doing next Wednesday? Because I am going to the game.
Also not sure because it's only 5.
Can you check when you are on campus?
Just remind.
Because I want to make sure I get whatever we're doing.
It's on my iPad.
Wait, next Thursday?
Wednesday. They're doing the game and I need to get pictures of for you before I fail that class.
What do you think you're gonna fail in?
Okay, well, I'm not going to fail that class, but I have to get 3 pictures in. Picture days in before the end of the semester. So I'm trying to knock them all out and then get extra credit to give that to an A. Because it's an A minus right now.
Alright, so we have DNA, and then we have a primer, and the DNA, the primer goes on the DNA to kind of say, okay, this is where replication is going to start. And then once the primer is on there, then we have our DNA polymerase that is going to read the strand. Here's our original parent strand. The parent strand gets split into 2 by helicase. The place where it actually is split is called replication fork. And then we have the original strand, which is blue in this case, and the new strand that is being made is pink. Here's our DNA polymerase as well. And then we have our completely new strand that has already been replicated here at the bottom. It's important to remember that these are identical, exactly the same. That's the whole point of this replication process, is to make 2 strands that are exactly the same as the original. So our original and this one, all the same. That's the whole point. Everything's the same. So just another way of looking at it, here's our original one and here's the result. The original one was the blue, and it ends up with 1/2 blue and 1/2 completely new yellow. And so that's why it's called semi-conservative, because we have the half is the blue, and then which is the original, and then half is new, so semi-conservative. When you think about it, it's kind of interesting because we all know we came from an egg and we came from a sperm, and that joined together, but then all of the rest of our cells in our body came from that one original cell, and then that split into 2, and then those 2 split into 4, and so on and so on. So one strand of DNA split into 2, and then these 2 strands of DNA split into 4. So somewhere there's an original strand of DNA in our body. Probably though at this point in our life it has degraded by now because it's probably too old and it's probably been reabsorbed into our body, because that does happen once DNA gets too old. So the original strand of DNA probably isn't there anymore. But there are other strands of DNA in our body that are probably pretty old because this replication process has gone on for a while. But like I said, eventually they do get degraded over time, and they do get replaced by the semi-conservative process. But it's kind of interesting to think about that there are some strands in our body that have been there for a while. Just another way to visualize it, this is in Google Classroom, but this also has descriptions that go along with it. Don't forget, as you are starting to study for tests, that Google Classroom does have a lot of good slides that I think a lot of people forget to go back and check the student notes section, but each chapter has a student notes section with slides like this that I think are really good summaries. And so if you are going back and studying, there's a lot of good stuff in there. Not only in there, there's other ways to study too. Another important thing to remember is that when the copies are being made, the DNA strand can only be read a certain direction. So I kind of drew it on the board. The DNA runs in opposite direction. There's something called a 5' and a 3' end. And the 5' and 3' go in opposite directions. DNA can only be replicated in the 5' to the 3' direction. So make sure you do write that down in your notes, that DNA can only be replicated in the 5' to the 3' direction, because that's a really important idea. Because as we're going to see when we watch the videos, it causes problems with the replication process. One side of the DNA can be replicated very, very smoothly, but the other side of the DNA has to be replicated in chunks. And these chunks are actually called Okazaki fragments, which we'll see in the video as well. By replicating it in these chunks, it makes it much more complicated. It can't be replicated really, really smoothly. These chunks are going to cause issues, And most of the time our body is able to compensate for that because our body's been doing it for a very long time. But sometimes by replicating it in chunks, it can cause mutations and other things to occur. But the reason it goes 5' to 3' and then it runs the opposite way is that our strands are just kind of like going reverse to each other. They're going backwards.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously. The DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
So as you can see from all that talk of like Okazaki fragments and things, there's a lot of room for mistake because it's being built in chunks. And so if something goes wrong with one of those chunks, it's very easy for a mistake to happen because it has to seal gaps, it has to go back and replace things, and so mutations can happen. Thankfully, our body usually catches the majority of the mutations, and it can repair them. So if a mistake does happen, our body can fix it. But if it doesn't, the other option is that our body can destroy the cell. It's better to lose one cell. I mean, think how many cells we have. Our body isn't going to miss one cell. It's better to destroy the cell than to let that one cell continue and replicate, which would cause more issues if a messed up cell was allowed to continue. No, don't start again. Okay. So it goes from the 5' to the 3' direction, and that's the way that DNA synthesis is another term for replication, is always going to go. One new strand of DNA is constructed in a single piece, and this is called the leading strand, and then the other is called the lagging strand, and it's created in these chunks, and the chunks were called the Okazaki fragments. I'm sure Okazaki, somebody figured it out, and that's why he got to name them after himself. Really, we all need to go discover something cool just so we can name it after ourselves and be immortalized forever in science. Darian, you're on your book, right?
Mhm.
I won most homework again.
I don't know.
I won most homework again.
Courtney has it. Ask her.
I think I won most homework again. I can't ever win anything fun.
Actually, I could check right now.
Oh, can't I win like most inspiring or most likely to win a Nobel Prize or something cool like that? No. Most
we have
homework
those?
given. Yes.
Didn't know that.
Yes. But no. I had to get most homework again. That's what I won last year.
Well, Miss Hayscam got the megaphone,
The what?
the megaphone person,
Yeah, that's what
and Miss Henry was like, I bet she only got it because she has to do
the lunch thing. Yeah. It's funny. Always fun with the homework.
I don't know. All I know is Miss Hemi and Miss Coach. I can't call Miss Director. Got social media. And,
I don't want to know what the others are. They should send out an email to us teachers so we know. Never anything cool.
During DNA replication, one of the two DNA strands, the leading strand, is replicated continuously, or all at once, in the 5' to 3' direction. The other strand, called the lagging strand, is replicated discontinuously, or in pieces, in the 3' to 5' direction. This is necessary because DNA polymerase, the enzyme that synthesizes a new DNA strand using a template strand, can only add nucleotides to the 3' end of a polynucleotide strand. Overall DNA replication is semi-discontinuous. To begin DNA replication, a helicase enzyme attaches to the double helix and unwinds the two strands, exposing them as templates for replication. The point at which the two strands are being separated is the replication fork. The enzyme primase attaches to each of the template DNA strands. Primase synthesizes a short RNA primer on each template strand. The primers are assembled in the 5' to 3' direction. After the primer reaches a certain length, primase drops off of the template strand. A DNA polymerase molecule attaches to each DNA template strand at the 3' ends of the RNA primers. Each DNA polymerase begins adding DNA nucleotides to extend the primer in the 5' to 3' direction. As the replication fork opens, the DNA polymerase on the leading strand template continues to synthesize a new DNA strand. However, the DNA polymerase on the lagging strand template, which can only synthesize a new DNA strand in the 5' to 3' direction, is moving away from the replication fork. Therefore, primase must again make an RNA primer on the lagging strand template, and DNA polymerase must continue making DNA beginning at the new primer. This results in a series of short fragments, known as Okazaki fragments, on the lagging strand template. The Okazaki fragments on the lagging strand template must be connected to form a complete DNA strand. To accomplish this, a different DNA polymerase recognizes and removes the RNA primers and replaces them with DNA. However, the polymerase cannot form the bond between the DNA nucleotide at the 3' end of one fragment and the nucleotide at the 5' end of the adjacent fragment, so there is a nick in the DNA strand. The enzyme DNA ligase is needed to form this bond and eliminate the nick. The process of continuous DNA synthesis on the leading strand template and discontinuous DNA synthesis on the lagging strand template continues until the replication fork reaches either the end of the chromosome or another replication fork moving toward it in the opposite direction.
So the top strand in this case is the leading strand, and it's really easy because it's just, as the helicase unwinds it, it just keeps going along without a problem. But because this one has to go this way, it has to wait for it to unwind, and then as it unwinds, it has another primer, makes a chunk, as it unwinds, has another primer, makes a chunk. So it's much more difficult, and it has that discontinuous replication process. Alright, so you have a new assignment that's in Google Classroom called the Replication Practice. Get you asking questions and thinking about how the replication process works. And the whodunit assignment is due today, I think?
No.
Do we make it due next week?
Yeah.
Okay, I couldn't remember.
When was that posted?
Earlier this week.
I never saw it.
Whodunit assignment due next week, extra credit due next week, and Strawberry Lab, when do we make that due? Due next week, due today?
Oh,
I don't care. That's fine, it can be due today. My main concern is as long as everything is in by next Friday. As long as I'm getting everything in by next Friday, that is my main concern at this point. I want everything in by next Friday. Questions?